Hello everyone, welcome back to the class. In our previous class we had learnt about peroxisome, which is another important organ discovered by Christian de Duve. It is a single membrane bound small organelle present in all eukaryotes. It produces and consumes hydrogen peroxide. They contain oxidative enzyme catalases and oxidases. The peroxisomal oxidase has a particular function. The major function of peroxisomal oxidase was, yesterday we had discussed this. So the oxidase transfers hydrogen atom to the molecular oxygen, and this forms hydrogen peroxide. So the peroxisomal oxidase helps in the formation of hydrogen peroxide. There is another key enzyme, that is catalase. Catalase uses this hydrogen peroxide to what? To oxidize varieties of substrates such as formic acid, formaldehyde, alcohol by peroxidation reaction. When there is excessive amount of hydrogen peroxide accumulated in the cell, this catalase converts this hydrogen peroxide into water. And we also discussed that the peroxisome lack DNA and ribosome. All peroxisomal proteins, that is peroxisomal matrix protein, peroxisomal membrane proteins, are encoded by nuclear genes synthesized on ribosome present in the cytosol and then incorporated into the pre-existing peroxisomes. So the ability of peroxisome to divide in a binary fission suggests that it has been originated by endosymbiotic association, which is similar to the mitochondria and chloroplast. So the localization of peroxisomal proteins to the ER and the similarity of some peroxisomal proteins to those localized in ER suggests the alternative hypothesis that peroxisome has developed from the ER, which is known as de novo origin. Most peroxisomal membrane proteins are made in the cytosol by membrane-bound ribosomes of ER and then integrated into the ER membrane, from where they may bud in specialized peroxisomal precursor vesicles. So new peroxisomal precursor vesicles may then fuse with one another and begin importing additional peroxisomal proteins synthesized by membrane-free cytosolic ribosomes to grow into mature peroxisome. So this image explains how peroxisomes proliferate and buds off from the ER and becomes a new peroxisome. So the major events occurring in peroxisomes are beta oxidation, photorespiration, glyosylate cycle. There is another specialized form of peroxisome, that is glyoxysome, which is present in some plant cells, mainly in the cells of germinating seeds, where they contain enzymes of glyoxylate cycle, which helps to convert stored lipid into carbohydrate so that it can nourish or it can provide energy for the growth. So today we will be learning about targeting of peroxisomal proteins from cytosol to peroxisome. It is easy and it is very much simple in the translocation or targeting pathway. So the protein is synthesized by membrane-free ribosome. So first, the proteins is synthesized by the cytosolic ribosome. So the cytosolic ribosome is first synthesizing the required proteins. The transport of proteins from cytosol to peroxisome occurring in post-translational way. So the cytosolic ribosome will transport the protein or the proteins which are in the cytosol is transported to the peroxisome post-translationally. Peroxisomal proteins synthesized on the cytosolic ribosomes are generally folded into their mature conformation in the cytosol. So the cytosolic ribosome, the proteins which are synthesized by the cytosolic ribosome is generally folded into their mature conformation in the cytosol before import into the organ. So the proteins that are involved in peroxisome biogenesis. So we know that peroxisome, to organize an organelle into a peroxisome, so they need some precursor peroxisomal proteins. So the proteins which are necessary for peroxisomal biogenesis and peroxisomal generation, and also proteins are needed for division of peroxisome, and some of the membrane proteins and also matrix proteins are all present in the cytosol. All these proteins are imported into the peroxisome. So the proteins which are destined or which are to be translocated to peroxisome, they contain a particular sequence that is known as PTS, which is known as peroxisomal targeting sequence. There is particularly two known sequence, that is PTS1, PTS2. So the protein which are to be translocated to the peroxisome is having particular sequence what? PTS, peroxisomal targeting sequence. So peroxisomal targeting sequence 1, PTS1 containing protein are recognized by cytosolic receptor PEX5 and are imported into the peroxisome in their fully folded form. There is another protein which is having PTS2 sequence, which is recognized by PEX7 receptor, and it is internally recognized and internally imported by this soluble receptor. See here, the PEX3 is a receptor inserted into the ER membrane. Pharecylated PEX16 from the cytoplasm help in the budding off of the pre-peroxisome. Then PEX16 and PEX3 mediates the import of membrane protein. PEX5 and PEX7 mediates the import of matrix protein. So this is nascent peroxisome, where after that, with the mediation of PEX5 and PEX7 receptor, the proteins are embedded inside, and it helps in expansion by continued import of matrix and membrane proteins. That expansion will lead to division of peroxisomes. So now we will learn about another important major central organelle of every cell, that is nucleus. It is a controlling center of eukaryotic cell. It contains the most genetic material of cell. Most eukaryotes have single nucleus and they are known as uninucleate. Some have many nuclei and they are known as multinucleate. Certain cells like RBC lack nucleus. Most nuclei are spherical, but multilobe nuclei are also common, which are found in polymorphonuclear leukocytes or mammalian epididymis. So the nucleus in the G0 phase has 4 components or 4 parts. They are nuclear envelope, nucleolus, nuclear matrix and chromatin. Nuclear envelope. It consists of 2 concentric membranes, that is outer membrane and inner nuclear membrane. Outer nuclear membrane is continuous with ER containing ribosomes bound to its cytoplasmic surface. Space between outer and inner nuclear membrane is known as perinuclear space, or we can say internuclear space. Inner nuclear membrane carries unique proteins that are specific to the nucleus. Function of membrane is that it acts as a barrier that separates contents of nucleus from the cytoplasm. Nuclear envelope contains nuclear pores or the holes for transport of macromolecules between cytoplasm and the nucleus. The nuclear pore consists of a multi-protein complex which are octagonally symmetrical and they have cylindrical structure. The inner and outer membrane fuses with the nuclear pore complex. Nuclear pore complex is having 30 different proteins known as nucleoporins. The proteins that make up the nuclear pore complex is known as nucleoporins. Nuclear matrix is another important component of nucleus, which is also called as nuclear scaffold. The network of protein filaments in the nucleus which provide a structural framework for organizing chromatin. Where exactly this nuclear matrix is found? Here you can see nuclear matrix. Nuclear matrix. So it facilitates transcription and replication. The ultrastructure of nuclear matrix consists of nuclear lamina, internal nuclear matrix. So nuclear lamina is nothing but the network of intermediate filament which helps in mechanical support to the nucleus. It links the internal nuclear matrix to the nuclear membrane. Nucleolus is another most important component, which is a non-membrane bound dynamic body. It disappears in the late prophase and reappears in the telophase stage of division. Each nucleolus is produced by nucleolar organizing region, which is known as NOR, and it is the site of transcription of ribosomal RNA, which is rRNA, and assembly of ribosome. It consists of 3 major regions. One is fibrillar center, second one is fibrillar component, and another one is granular region. Fibrillar centers contain rRNA genes in the form of partly condensed chromatin. Fibrillar component surrounds the fibrillar centers which contains RNA molecules in the process of transcription. Granular regions are the outermost regions having mature ribosomal precursor particles. Nucleolar organizing region. As I said, so nucleolus is produced by the nuclear organizing region of the chromosome. So it is the regions of chromosome around which the nucleolus forms. In humans there are 5 chromosomes containing NORs, which can be identified as secondary constrictions on metaphase chromosome. Here you can see the metaphasic chromosome. As compared to secondary constriction, the primary constrictions are also known as centromere. This is the primary constriction which attaches the sister chromatids together. The difference between these two constrictions are noticed during anaphase, as chromosome can bend only at the site of primary constrictions. The area of the chromosome distal to the nucleolar organizer is called satellite and trabant. The chromosome with the secondary constriction usually associated with the nucleolus is called SAT chromosome. So each of this structure can be visualized during the cell division, and You will be learning about this in detail when I start cell division. So in our next class, we will be learning about transport of proteins through nuclear proteins. Thank you.